Imagine a guided missile zeroing in on targets. That's what modern cancer drugs aim to do. Extract tumor cells while sparing healthy tissue. Sounds like a science fiction. It's real and it's saving lives every day. Cancer used to be treated with the blunt force tools like chemotherapy and radiation, carpet bombing the whole body. But today, we have precision weapons. How did they discriminate friend from foe? Normal cells from cancer cells. Let's dive in. Most targeted cancer therapies exploit molecular difference between malignant and healthy cells. Here's the main strategies. Tumor cells often overexpress specific surface proteins receptors that signals them to grow. Drugs can bind these receptors like a key in a lock, blocking growth signals or marking the cells for destruction. Trastuzumab, for instance, attached to the HER2 receptors in a certain breast cancer, flagging cells for our immune system to attack. Inside every cell, proteins called kinases regulate growth by turning other proteins on or off. Cancer hijacks these kinases, running growth signals non-stop. Small molecules like imitinib sleep inside cells and block those rogue kinases, effectively pulling the plug on tumor proliferation. Think of ADCs as Trojan horses. A monoclonal antibody hones in a tumor antigen and is loaded with a toxic payload. One internalized, the drug is released and it kills the cancer cells from within. Some cancers, like many breast and prostate tumors, depend on hormones. Drugs such as tamoxifen or androgen receptors blockers starve the cells of the hormonal signal they crave. In each case, the drug design hinges on a molecular signature unique to cancer cells. By locking on that signature, the therapy achieves specificity, and that's the cornerstone of precision oncology. At the first glance, it feels like we've solved the puzzle, match a drug to a cancer marker, and the disease disappears. But biology isn't that neat. Tumors are not uniform. They're messy, shape-shifting ecosystem. A single biopsy may show one mutation, while another part of the tumor hides a different signature altogether. And cancer cells don't sit still. Under pressure from therapy, they mutate, adapt, and sometimes borrow tricks from normal cells to evade attack. This means even the most brilliant smoke bomb can lose its targets over time, setting the stage for resistance and relapse. Precision doesn't mean perfect. Cancer cells are clever. They mutate, evolve resistance, and sometimes mimic healthy cells, blurring the lines. Off-target effects. Even the most selective drugs can bind similar proteins in healthy cells, causing collateral damage. Resistance mutations. Tumors can acquire new mutations that prevent drugs binding, effectively closing the lock. Immune-related adverse events. Checkpoint inhibitors can unleash harmful inflammation against normal tissue, leading to colitis, pneumonitis, or endocrinologies. When you start a targeted therapy, you might see fewer classic chemo side effects like hair loss, but new ones emerge. Skin reaction, rashes, or dry skin from EGFR inhibitors. Gastrointestinal options, nausea, diarrhea, and most cells from small molecule drugs. Infusion reactions, fever, chills, or low blood pressure during antibody infusion. These often appear within days to weeks of treatment. Most are manageable with supportive care. Antihistamines, typical steroids, or dose adjustments, but they remind us that even targeted therapies have systematic footprints. In the long run, precision drugs can leave a legacy of chronic issues. Cardiotoxicity. Agents like trastuzumab can weaken heart muscle, leading to heart failure years later. Bone marrow suppression. Some kinase inhibitors impair blood cells production, causing anemia or infections. Secondary cancers, very rarely, DNA-damaging payloads in the ADCs or off-targeted kinase inhibition could increase the risk of new malignancies. And then there is a resistance. Tumors may initially shrink, only to regrow with the new genetic alternation, demanding next-generation inhibitors. So how do cancer drugs know which cells to attack? They target molecular flags that cancer cells either produces in excess or uniquely present on their surface. The precision approach spares healthy cells at least more than traditional chemotherapy does and has revolutionized oncology. Yet, these treatments come with their own hidden danger and evolving challenge. Tumor adapt, side effects persist, and the long-term safety remains under close watch. We are part of a grand experiment, pushing the boundaries of what possibles, one molecular target at a time, as science discovers new cancer signature. Tomorrow's drugs will be even more selective and hopefully even safer. For now, every targeted therapy is a trade-off, a powerful weapon against cancer and its own risks and rewards. Is it perfect? Not yet. It's a masterpiece of a molecular engineering and a treatment to how much we can achieve when we learn cancer secrets.